Biodegradable polymers are polymers that degrade via chemical, biological, or physical interactions with microorganisms in the environment into non-toxic byproducts, so no microplastics. Biodegradable polymers are normally degraded via bonds which are easy to hydrolyze or bonds which are sensitive to enzymes. Here are two biodegradable polymers, polylactic acid and polyhydroxybutyrate. They both contain ester links, which are easier to hydrolyze than hydrophobic carbon-to-carbon bonds as seen in saturated polymers such as polyethylene. They have shorter aliphatic chains between ester links, which aid degradation. So why do we need biodegradable polymers? There is an abundance of plastic in the environment, which can cause many problems. Plastic can block pipes and sewers, which creates breeding grounds for pests, and in turn increases transmission of vector-borne diseases. Also, many animals digest plastic, which can block the airways and stomachs of wildlife. An OSPAR monitoring program found that between 2014 and 2018, 92% of northern farmers had ingested plastic, and 51% had ingested more than 0.1 grams of plastic. This map from the program shows the proportion of northern farmers with more than 0.1 grams of plastic in their stomach. When wildlife ingests plastic, it can cause toxins to enter the food chain. Polymers containing benzene or styrene are carcinogenic, and ingestion can cause problems for the lungs, nervous system, or reproductive organs. Biodegradable polymers reduce the amount of plastic stuck in the environment, causing these problems. How do biodegradable polymers biodegrade? This happens in 3 steps: biodeterioration, biofragmentation, and assimilation. Biodeterioration is a surface-level process. The microorganisms grow on the surface and affect the polymers in physical, chemical, and enzymatic ways. The microbes secrete a complex slime matrix of polymers, which accelerates the biodeterioration. Water enters the matrix, encouraging hydrolysis of polymers. Biofragmentation involves microorganisms secreting enzymes or free radicals to break polymers down into smaller fragments, so monomers, dimers, or oligomers. The enzymes act as catalysts to the breakdown of polymers. This can be done via radical oxidation, enzyme oxidation, or enzyme hydrolysis using water from the biodeterioration step. During assimilation, the fragments permutate the cell membranes and oxidize to ATP and intracellular structures via aerobic respiration, anaerobic respiration, or fermentation. In order to form the intracellular structures, the microbes use the fragments as sources of energy, electrons, and atoms. These processes allow microbes to grow and reproduce. This also produces small organic molecules and minerals, which contribute to biogeochemical cycles. How are biodegradable polymers used? There are many industries that use biodegradable polymers. For example, agriculture, electronics, packaging, construction, and medicine. I will talk about the applications to medicine and agriculture. In medicine, both natural and synthetic biodegradable polymers have many uses. Biodegradable polymers are useful as they can degrade into the body once their use is ended without needing to be surgically removed. For natural polymers, proteins get used for tissue engineering, drug delivery, and structures due to their compatibility with the body. And chitin has also been used for wound dressing and drug delivery. Biological implants are often made from synthetic biodegradable polymers as there is more control over shape and properties. PLA and PGA are considered the first synthetic biodegradable polymers used in medicine. Here is an extract from a table displaying PGA, PLA, and their monomers. PGA is made from glycolide. PLA is made from lactide. Lactide has 3 isomers, 2 optically active isomers, L-lactide and D-lactide, and 1 optically inactive isomer, mesolactide. L-lactide is naturally occurring and can polymerize to PLLA, poly-L-lactide. An amorphous polymer, PDLLA, poly-D-L-lactide, can also form but has a lower tensile strength due to its lack of organized structure. PGA and PLLA both have high crystallinity. PGA has crystallinity of 45 to 55%, and PLLA has crystallinity of around 37%. This means they have good mechanical properties, but they have very different degradation times. PGA degrades within 1 to 2 months, but PLLA degrades in 2 to 5.6 years. Both degrade into compounds compatible with the human body, glycine and glucose. Glycolide and lactide can copolymerize to PLGA, which can be constructed with different proportions of each monomer to form polymers of different tensile strengths and degradation times to suit different biomedical purposes, for example tissue engineering and structure reinforcement. In agriculture, starch can be used for slow-release fertilizers. Starch is a naturally occurring polymer which is widely used due to its biodegradability, abundance, and non-toxicity. But as native starch, it has low mechanical properties. It can get modified to alter its properties, for example by cross-linking. Cross-linking uses the hydroxyl group on the starch repeating unit and a cross-linking agent, for example citric acid. Citric acid forms hydrogen bonds with the hydroxyl groups in the starch molecules. This changes the hydroxyl group to an ester group, reducing water vapor permeability. This allows it to be used for slow-release fertilizers. Starch coats urea, a fertilizer used by farmers, as a semi-permeable or impermeable membrane, which is a physical barrier separating urea from the soil. As the cross-linked starch degrades, the fertilizer is released. This acts as an alternative to non-biodegradable coatings which contaminate the soil. Why don't we rely on biodegradable polymers? Lots of plastic advertised as biodegradable either takes years to degrade in several natural environments or degrades into microplastics, which isn't an improvement. Here are graphs from a study on biodegradability of plastic bags, showing the tensile strength of different types of plastic bags after time in the environment. The bags labelled as biodegradable had very similar levels of biodegradation as conventional plastic bags, showing that they don't reduce the environmental impact of plastic bags. Both oxo-biodegradable bags were found to degrade into microplastics, which are harder to remove from the environment. This shows that biodegradable plastics still require development before they will be a complete replacement for plastic. In the future, biodegradable polymers will need to more effectively biodegrade. This could be done by finding microorganisms which can degrade polymers of higher molecular weight. Also, being able to pinpoint how long biodegradation takes would be beneficial to many industries such as medicine. Improving their properties in general would give them wider application to everyday life, and by improving synthetic techniques for biodegradable polymers, it would make them cheaper and easier to produce, making them more widespread.